All right, it's a pleasure to introduce our next speaker, Professor Brian Wandell, who's been a colleague of mine for the past 24 years or so. Brian is the Isaac and Madeline Stein Family Professor in the Department of Psychology. He's also, by courtesy, on the faculty in electrical engineering, ophthalmology, and the Graduate School of Education. Brian has been at Stanford since 1979. And during this time, in addition to all of the amazing science that he's done, he's been a leader within our community in many different ways. One way is he's the founding director of Stanford's Center for Cognitive and Neurobiological Imaging, which has had an impact on many of the labs across campus. And he's one of the founding deputy directors of the Tsai Neurosciences Institute, being a deputy director from the beginning, 2013 to 2022. His research centers on vision science, spanning topics from visual disorders, reading development in children, digital imaging devices, and algorithms for both MRI and digital imaging systems. Those who know Brian's work in the MR space know he is a leader in functional, structural, and quantitative MRI. He's built numerous important software tools for MR data analysis and data visualization. In terms of vision science, he's advanced the understanding of the organization of visual field maps in the human brain, color and motion processing within these maps, the potential for map reorganization, and the development of cortical circuitry for reading. He's an elected member. Yes, I'm winding up, Brian. Elected member of American Academy of Sciences. You get to an age where you don't, I guess, want, I don't know, Brian, you want me to wind up? As member of the National Academies, Brian.
Thank you, Anthony, and it was very kind of you, and enough is enough, so thank you. And thanks to all of you for being here. I'm excited to be able to tell you this story. I'm going to take you through actually 150 years worth of work, not all my own, you know. And really it's going to be divided into three parts. One is I want to talk about the human visual system and the visual field maps, retinotopic maps. Second, I'm going to spend some time on something that really relates to what a number of the other speakers have talked about, which is the plasticity and stability, both, of the human brain. Those are both important and happen in different places and times in different ways, worth understanding. And the last little bit, I'll take you quickly into the question of something that I really was ending my work in MR on, which is the ability to diagnose reading disorders. And people have difficulty in learning to, particularly to see words. I'll tell you where we got on that, and that connects to some of the work that, I didn't know that about you, Jay, but some of the work that Bruce was talking about earlier on today. So, you know, our understanding of the visual parts of the brain started really around 1860. And there was actually a huge fight between one of the guys shown here, this guy Monk, and Ferrier, Sir David Ferrier, who one said, well, it's way at the back of the brain, and the other said, no, it's in the middle of the brain, and they fought one another over this, and the wars were really like Twitter wars. I mean, it was really quite brutal when you read it. People wrote their papers with much more directness in those days. And it wasn't really resolved. Monk was completely right, and Ferrier, who we honor and so forth in many lectures, was completely wrong. But what really resolved it was these tragic events of war, and particularly the Russo-Japanese War and the Great War of the World War I, where people could, bullets would be shot through their brains and skulls, and they would lose sight, but they wouldn't be killed. And these folks here, Inoue and Holmes in particular, along with some Henshin, but mostly Inoue and Holmes, realized, wow, in the back of the brain, where Monk had identified visual cortex to be, and that's what's shown over here. You're just seeing a little bit of the back of the brain. I'll do more for you in a moment. On the back of the brain over here, you could see that if they had a bullet through here, they lost the ability to see in the central visual field, whereas a bullet here or here, they would lose abilities in corresponding parts of the visual field. So the visual field maps were identified for the first time in human about 1900, 1910, something like that. So there were then many years of the rise of electrophysiology, the folks who would take electrodes and place them in the brain and so forth. And starting, in around 1940, it was realized, wow, there's not just one of these organized maps, but there's many. Every week, I remember as I was a graduate student, every week another one would be turned up and we'd all be excited. And I'm guessing that Kong and Corey and others here, Anthony, have all seen this particular picture, this map from Fellman and Van Essen, which was an early attempt to summarize all of these different maps in different ways and kind of stuck in the mind of the field. These were all based on animal models. And even at that time, in 1991, we really didn't know much to what extent human was the same as any of these, even primate models. We didn't really know. And so these very distinguished folks here, Jonathan Horton and Bill Hoyt, said, yeah, we just don't know even about the second map. OK, so why didn't we know about human? Well, we couldn't really measure human. And in fact, many of the animals were different from the models, were different from themselves. And the technology that we had then is around 85, 90, and so forth, is shown here as PET, and people were excited about it. And this was what would get you a Nature paper in 1986. You could take 5 subjects and have them stare in the fovea and then put something out in the periphery, and you'd see this white spot over here moved from here to here, and 3 or 4 pages later and a bunch of analyses, you had yourself a Nature paper. So from that, however, around 1990 was the rise of functional magnetic resonance imaging. And that was the first time that we could really peer into the living human brain and see the activity there. And in fact, over that 30 years, and this is what Anthony was measuring, our group, but many groups, all contributed, sometimes fighting, sometimes helping, sometimes this, sometimes that, contributed in developing tools so that we could, again, here's another picture of the posterior part of the brain segmented, and we could, at that point in time, this is from a review article with John Winnower, we could say, well, here's primary visual cortex with this resolution in the human brain. And the second map surrounds it, V2. And the third map surrounds that, and also that's the third visual field map. And we could find them, and now we find them in experiments that take about 10 minutes in an fMRI scanner, and we can localize them and know them. So what is it that we did, and how did we find that out, and what else did we learn besides confirming the basic organization? Well, the first idea, this came from Steve and our group here, a bunch of people here were involved. But if you stare here at this red fixation and that stimulus expands like that, you can actually, in the functional magnetic resonance imaging measurements, actually see a kind of traveling wave go right through posterior part of the brain here. And that really is mapping for you the eccentricity map. Notice, by the way, while we're here, there's just a little bit of stuff coming on down here. Not main point, but we're going to come back to it in a moment. And so you can represent that the central visual field is represented here, and then you can see the colors red to green to blue to violet, red to green to blue to violet, and you can measure it, and you can learn things about the basic organization. The way you find that there are multiple maps is you take the stimulus and you rotate it instead of expand it. Okay, and then you get these rather surprising findings that this part, well, not surprising, this is how you know what the map is. This part, red, is responsive when the stimulus is here in the lower visual field. This part, blue, is in the upper visual field. So this is a whole hemifield, a half of a field. It's the right hemisphere, so it's the left half of the visual field. And then it flips back to green, and then it flips back to blue. And that's how you find the other maps, the V2 and V3 maps. Okay. What was completely new and surprising to us, and not really part of the animal literature as we started to see this at scale, because we had living humans, we had a large part of the brain. It's hard to do these things with microelectrodes. You're happy when you move 1 or 2 millimeters, but here we are going multiple centimeters. You could see that there would be these other parts that were representing the fovea over here and also expanding on out. So here was one part that we're looking at. This is the primary visual cortex, V1, V2, V3. Here's another region here with another little fovea and another little map. And in fact, if you look at the brain broadly, I'm just going to take you through 15 years of work. This is the first one that we were looking at. This is the one I was just showing to you on the ventral occipital part of the brain. It's like a little fovea with a little expanding map. Another one here on the lateral side, lateral occipital, a little fovea and expanding map. And there are several more. This is around the motion selective cortex, a fovea, and an eccentricity map. And up here into the intraparietal sulcus, a couple more. And in the early days, people were naming all these little maps by these things like MT, MST, mumble, blah, 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 blah. It sort of looked like genomics or something. I had no idea what it was. And we couldn't remember all that stuff, so we started naming them by where they were. Lateral occipital, ventral occipital, and so forth. And I just want all the students in here to know that that happened, and you're welcome. Okay, so now once we had identified the maps, the next thing to do is try to measure the signals and quantify them coming through the different maps. And we really didn't do, it was no giant breakthrough, but it turned out to be influential. He said, look, let's look at the time course of these signals and let's build little models of the receptive fields of each voxel. And this is called population receptive field. So you can say, well, if I've got a voxel here in the brain that I'm measuring, a location in the brain, and I move the stimulus around, can I predict how this voxel will respond? And that turned out to be very informative. In primary visual cortex, the regions of the world that would drive a particular location in cortex were quite small. And if you went around to the motion selective areas, they were actually quite big. And we could measure them. What are shown here, a bunch of curves. Primary visual cortex is this curve. It's from the fovea to the periphery. It rises a little bit, but it's kind of varies quite a bit across these maps. And we could model that and so forth. And then there's been a whole set of follow-on work by the people shown here and many others to build quantitative models of if you put on a stimulus, what response will you get out of each one of these maps in an effort to try to understand what it is they're doing. And there's really quite some consensus about the maps, which are not quite like the macaque model, honestly. There's noticeable and quantitative differences and completely unlike anything that you might find in a mouse. It's completely, not at all the same, maybe not surprising, but it's a big thing. So there's certain things you can learn from mouse and other things you can't. Okay, so with that in mind, as we were developing this, I became quite interested in the question of plasticity. Just as Marion was talking about plasticity for stroke, there are many ophthalmological diseases, retinal diseases in particular, in which you lose visual function. And at that time, as I was coming up to speed here, there had been kind of a dominant thought in the literature from the Wiesel and Hubel that there was a critical period and that things could very adaptable early on, but then not so adaptable a little bit later. And then down the road, we had Mike Merzenich, who really had a big impact on MTL and others, and John Koss. And they were going, oh man, plasticity. You couldn't turn on NPR without hearing an advertisement of how about the brain is plastic, Oh my God, we're all gonna live forever. So I was excited and interested in this, and various things kept coming across our lab's plate. I was fortunate to be surrounded by a bunch of really just great postdocs. And one of the things that happened, I'll tell you a series of little vignettes about that and then come to a rough conclusion. One of the things that happened is, you may not know, but you will in about 30 seconds, that the axons that come out of the nasal part of the retina, so here's the retina, the eyeball, the blue stuff over here is near your nose, so the nasal part, that crosses to the other side. And here's the nasal part in the other eye, it crosses to the other side. The temporal part, shown here in blue, stays on the same side. The temporal part stays on the same side. The result is which you all have, pretty much every one of you, I can guarantee, in this room, that's the organization of your visual pathways. Okay, and you can see this in structural MR. It's not that hard. You can see the crossing there. That's called the optic chiasm. And a friend of mine, Joyce Liao, who's in the ophthalmology department and a great clinician and scientist, called up and said, could you check this, Brian, because we got this guy. I say, oh, okay. Well, that's no chiasm, right? There's just missing optic chiasm. And it turns out that people who are achiasmic like this are, I don't know, 1 in a couple of million. It's not that many. But, we're doing more and more scans, and you run into this stuff. Okay. So we thought, well, okay, this person, by the way, this person didn't come in because they had vision problems. They had some headaches and had some other issues, but they could see just fine. And the individual could play baseball. He wasn't that great, but he could play baseball. He could read. He could do all the kinds of things that you normally think of. okay? And so she asked us to run some checks. We did a series of studies, including, well, okay, you can see this eye is blocked off to make the experiment easier. And then we put stuff in the left visual field, and sure enough, the right hemisphere responded. And the colors here are indicating kind of one of these retinotopic maps. And then if we put things in the other visual field in your brain, it would go to the other hemisphere. But in this guy, it stayed in the same hemisphere. And so as expected from the anatomy, you could see it in the structural anatomy. And then we could go and model, well, gee, if you look at a piece of cortex in this fellow, what's this person's receptive field? And it was quite interesting. It's shown right here down the midline, one side and the other side. These two sides here kind of, even though they're in the same place in cortex, two different parts of the visual field would drive it equally. And so it was as if the two sides of the retina, when they came up, folded on top of one another. So, and the fact that he could see and live like that, I just thought was incredible. And there was nothing really wrong with his basic vision. And so, though of course he didn't have stereo vision. That was clear. Okay, so that's a development, right? He's born like that. And we kind of know why and what happened. That's very impressive that during development, the ability to learn to see with that visual system is quite possible. And along with my good friend Nikos Logothetis and Stelios became my friend Spironakis, we said, look, let's try to get these kinds of plasticity. Let's deal with the Merzenich, Kaas, Hubel, Wiesel. Let's deal with this stuff a little bit. And let's see if we can get this under control and measure it directly. And Nikos had a massive shop in Tübingen. And so we would do things like put in lesions into the brain to see what recovery we got. And here we were shocked because we had been reading all this stuff about how things would change so much. And here we would introduce these lesions, and what you're seeing here is a macaque brain. V1 is on the operculum, the surface of the brain here. If we lesioned it, there was an absence of signal. We thought we were going to see this fill in, but actually we waited week after week after week after month, and there was a couple of hundred microns change, or maybe nothing. So for people who are studying the resolution at 50 microns, 100 microns, a millimeter, they were very excited when they would see a little change. But for us, we're looking at across a centimeter, it was basically very little, and it stayed like that, and that was a little dust up, but never mind about that. We then went on to look at lesions that we didn't introduce into the non-primate, but that were a consequence of disease led by, I was fortunate to have a group of ophthalmologists come from Tokyo over the years. It was a great group. And we looked at juvenile macular degeneration and retinitis pigmentosa and so forth. And it was roughly the same story, and the papers tell that story. The papers that we published on this tell that story. In the adult, when this developed, or when we introduced the lesion, or when the disease was in later years, the amount of shift in the peripheral representation, the plasticity, was not great. So there's tremendous plasticity in some cases, not much in others. By the way, this was viewed as great news for a lot of the people supported here, meaning Chichelnitsky, Palanker, all the others who were making retinal implants to overcome the problems with these diseases. They're happy to know that, in fact, the retina has not, visual cortex has not completely reorganized, that it's sitting there waiting so that when you put in a good signal with an implant, it's not as though you're fighting what the visual system has reorganized into. So the fact that there's no plasticity in this case is kind of good news for certain kinds of developmental fixes. Okay. I would say I just want to briefly mention one case in addition to these. This has a long history, and I'll just put these here. But we were, Cheselden was the first, 1728. I told you there was going to be stuff like this. So this guy here, Mike May, who's had books written about him and is quite public about this. Mike had lost his cornea and lens at the age of 3, and they couldn't be replaced because the stem cells couldn't be put in place, back in place. But after 43 years of living without contrast vision, Mike could have his cornea and lens restored. And we were fortunate to be part of the group. Ione, Netta Levine, our close colleague who's at the Hadassah, chair of now of neurology department in Hadassah in Jerusalem. We studied Mike, and in fact, even though he lost contrast vision at 3 years of age, and then had no contrast vision. And then it was restored by a proper corneal and lens transplant with the proper stem cells. His vision never really could recover. So we measured him year after year after year, and his cortex looks unlike your cortex and mine. And it's documented in the papers, but let's just say he really never got there. Wonderful guy, still uses a cane. He's got all kinds of technology. So again, when and where the damage is determines the plasticity, both there. Okay, last thing is I've spent quite a number of years doing my best. And I want to mention Jason Yateman, who's here on campus now in the Graduate School of Education and also in the med school, who's continuing this work, and I did a lot of it with Jason. We really had this hopeful notion that all these things we had measured about the maps and about the PRFs and about plasticity and about development and so forth would lead us to be able to do something helpful, kind of science that would help the reading community. And a review paper I wrote about what we had learned and others in 2017 is listed here. By the way, all these slides I always post my slides and make them available on my website. And that also has a long history from Wernicke and Jules Desjardins and Paul Broca and so forth. But the basic principle, the reason I put this picture here is the basic principle is that we don't think of reading as being localized. Reading isn't a spot on the brain. Reading requires being able to see the characters, encode them in the retina, bring them into V1. V1 then does something to it that'll distribute it somewhere else. And you can have a problem with reading because of the eye movement circuitry or because of V1 circuitry or because of reading specific circuitry or because of language circuitry or, and I would say most importantly, because you don't get a good breakfast in the morning because you walk to school and somebody might be shooting in your neighborhood. And so there's a lot of ways to fix reading. And we took our slice. I wish I could stop the shootings and other stuff, but we took the part we could, which is the part of, okay, let's see what we can do, because there are people who are quite well-to-do. We had early support from the Schwab family. A bunch of them can't read very well. And for them, the problem was not, not lack of money or lack of resources and so forth. So here's the story about reading, just for the last couple of minutes. On the ventral part of the brain, a little bit past where these maps are, but just adjacent to them, there's a number of places that have kind of extra large signals if you put on something moving or something with faces or something with color and so forth. And my colleague, Colonique Grill-Spector and Kevin Weiner, who's at Berkeley now, have done a nice job laying out in this review paper a few years ago what it is we learned about the relative distribution of these areas and so forth. And I'd always been a little reluctant to go into it, but the reading community was really obsessing over a particular region over here. They had the nerve to label. No, I don't mean that in a bad way. They had the guts to label the visual word form area. Okay, so they really wanted to, they took a thing. And so we thought, well, okay, let's see if we can see the signals going into there. And with Andreas Rauschecker, we would do things like put on words over here, and you could see the activity in V1 if the word is here just to the side of fixation. You could see the activity in V2, V3. Not surprising, right? You can see the activity in the fourth map, map ventral occipital, and boom, right there in the visual word form area. So we took that as, okay, well, that's good. You can actually start to track these signals as they go forward in the brain. This is two views, by the way, of the same person, but we get this. You could get this in many experiments. So we started to look at what the responses were in the visual word form region, along with other regions and so forth. And so I remember a moment ago I was telling you about these population receptive fields, and they so forth. Now the regions down there, that's how much of the visual field responds, will drive a response in this location in cortex. That's the receptive field. So we say, well, in these cases here, we have maps. So you can take all the voxels here, and they're laid out in a map, and you can see it nicely. But if you put all the voxels together, they tell you what the whole coverage is of that area. In these reading regions, there are no maps. So they're not organized in that same map-like way that it goes from fovea to periphery and so forth. But you can still do this. you can measure the population receptive field of all the voxels and assemble them all together, as I've done here for V1, V2, and V3, and say, well, how much is the coverage, overall coverage, in that region? And what we discovered, and this is Rosemary Lee and Gary Lerma, who is a fierce partisan for the Basque Country, and you can look at the coverage here. And in the reading portions, I'll show you 20 others, you get a coverage really only in the central visual field, only makes it into there. And I don't know if you've ever tried to read by putting something 10 degrees in the periphery or 15 degrees in the periphery, but you're not going to be any good at it. Okay? And it doesn't really matter if you make the letters really large. You're still not going to be any good at it. So we thought, well, okay, that's interesting. That's like what part of the retinal signal is making it to these regions specialized for this. And we had to look at a bunch of different kids, and they all have slightly different shapes. And we didn't quite, to this day, I'm not sure what to make of it because you have to think of the whole system. Your eyes move around also. So if a little bit makes it, well, it's got to be a combination of the eye movements and the coverage and so forth. And so the direction that we were going in, and here's Hiramasa and Franco Pestilli, and we spent a whole bunch of years in my life looking at these white matter pathways and the connections and sort of trying to understand that, and there's a series of references there about it. But the direction we wanted to go is, again, system work. We're not looking in a millimeter, we're not looking at 500 microns, we're looking at the connections between the regions, spot to spot to spot, what's the white matter connections, and we're working very hard to build quantitative models that start out with what do we know about the cornea, what do we know about the lens, what do we know about the retina, where do the signals go, and what can we do to diagnose the problems in individual kids so that we can tell that kid, you're going to be okay. You are going to need to get an assist because the reading is never going to go forward for you, but for you, it's going to be okay, and we can help the parents who need the help and tell the other parents, that sorry, we can't do much for you. I just wanted to take a moment to thank all the people who worked on this. I didn't get to mention each of them individually, but I've been so fortunate to be at Stanford. People have been thanking the Institute, and I'm grateful to the Institute. I'm also grateful for the many years at Stanford prior to the Institute. And as I was going through these, and looking at them, Michal Ben-Shahar is a dean at Bar-Ilan University, and Netta Levine, I mentioned, is chair of the neurology at Hadassah Hospital, and Aviv Mezer is a professor in the Hebrew University in Jerusalem. And these days, I have to say, as I look at these pictures, my heart goes out to them and to all of us for these challenging times. And it was, I'm hoping for the best for all of us. I also wanted to particularly call out these 4 ophthalmologists who, just sitting there minding your own business in the psychology department, and some dude calls you up from Tokyo and says, I'm an ophthalmologist, I'd like to come and work with you. And boy, was I lucky. They came, they were smart, hardworking, brought us into all this work on macular degeneration and so forth. I'm very grateful. It wouldn't have happened to me if I wasn't at Stanford, I have to say. So I'm very grateful to that, and thank you all for your attention.
Thank you, Brian. We have time for questions.
Thank you. A fantastic story. My question is about the future. What do you think are the next challenges in the field, and how complete do you think the maps are at the moment?
Yeah, thank you. The maps, I got out of the map business a while ago. I think there's tremendous consensus in the sense that multiple groups have repeated these measurements. Sometimes we worry in neuroimaging about replication and so forth. These have been repeated by many groups and many times. So we have a baseline, and there are great tools for quickly getting the maps. So the thing that I think, I spent a lot of time in the electrical engineering department, and I'm very impressed how groups come together with different skills when they're confronting bigger stories. And I think that these matters of building up the whole pipeline in understanding how it is the signals are flowing through, both in the white matter. I mean, we heard, Marion's talking about parasites, and before Ben Barris passed, we had started some projects with him on glia and so forth. The glia, the neurons, understanding the whole system through large integrated projects seems to me the future. And the past seems to me, oh, look, I have the spot here, and look, this is the spot for reading. And that was the past. The future, I think, is systems work and involves not just an individual, but involves collaborations between large teams. And I think that we're ready for that in this part. You already see that happening at some of the cellular work, and I think that's an important part for what we need to start moving towards in this kind of work. So thank you for the question.
Thank you, Brian. Could you say something about dyslexia and what goes wrong there and how people can learn past or not learn past dyslexia?
Yeah, so like so many other diseases, dyslexia is heterogeneous. The most diagnostic measure that we have of dyslexia is actually not a visual test at all. It's phonological awareness, it's called. And this is the ability to hear the distinct sounds of speech and to relate those to the printed text. So, and that's the thing that the field had really focused on. And when I first entered reading, I would show up at reading conferences because, go and see what the pros know. And they said, what's a vision guy doing here? Vision's got nothing to do with dyslexia. But over the years, the phonological awareness parts, the early prediction and the use of that. And so the notion was, well, if we train people on phonological awareness, then they won't, they'll get better. And that hasn't really panned out all that well. So people are now open to looking at other aspects of it and the visual parts. And maybe next year you'll have Jason Yateman here. And Jason has built on this story to say he's been looking at changes in the white matter and the pathways that connect visual cortex to auditory cortex and the speech areas. And we're just looking around for many different things like that. So I'm not giving you a crisp diagnosis, but a sense.
Great. Let's thank Brian again.